Alright, so this one's not going to be the best one because I don't have the board to write on, but I'll just quickly go through it. So genotype, remember these are the letters that you use, or the genes. The phenotype is the trait. And what we're trying to do is use a Punnett square to determine what is the probability that we get certain genotypes and phenotypes of our children. So there are two types of genotype. Alright, heterozygous, remember, means that we have a big letter and a little letter. And this is always gives you the dominant phenotype. Where homozygous means you have two capital letters that are the same, and that will give you the dominant phenotype or trait. Or you can have little letter, which gives you the recessive phenotype or trait. And that should be little letter. Alright, so that's heter. So, and then finally, remember we have allele. Allele is just a type of a gene. And we have the dominant allele, which is the capital letter, and the recessive allele, which is the little letter. And the recessive allele, in order for we to get the recessive phenotype, we need to have two of these, but the dominant allele always overrides the recessive allele. Alright, so you should remember those things, so those are just background. So now what we're going to do is use a Punnett square to figure out what the what we can get for the babies. So if we have a heterozygous tall person, so it doesn't matter where you put these on. I'm going to put this one at the top. I'm just going to use, actually, I'm going to use T's because that's going to be the easiest. So there's a capital T and lowercase t. Ignore that A. Alright, and then there's a homozygous recessive short person. So I'm going to put the short person over here. But again, it doesn't matter where they are. So you put one letter in front of the box, each box, and you put one letter on top of each box, and then you just fill it in. So this big T is going to go in both of these boxes because there's a big T above it. A little t is going to go in both of these boxes because there's a little t above them. And then the little t is going to go across these boxes. Alright, and this little t is going to go across this box. So now our genotype percentage is going to be 50% because half of them, two boxes, are big T, little t, and then 50% are little t. Again, two boxes of that. Phenotype ratio, I'm not really too concerned with that. Let's just give the percentages here. Well, actually, the ratio. Alright, so these two, because they have a big T in them, and sometimes I like to just circle all the ones that have a big T, those are going to be the dominant ratio, the dominant phenotype. So there's two of those, and then there's these two that are going to be recessive. So it's 2 to 2, or just like our genotype percentage is 50% and 50%. 50% dominant, 50% recessive. Alright, so the next one here, we have two heterozygous cats. So we'll put one heterozygous up here. That's a big T and a little t. We'll put one heterozygous over here. Big T, little t. Then we go and fill it in. So there's a big T all the way across. There's a big T all the way down. Then this little t goes across. And this little t goes across. Alright, so that gives us 25% big T, 50% big T, little t, and 25% little t. Our phenotype ratio, so if I go back and circle all the ones that have a big T, the phenotype ratio here is going to be 3 to 1 because there's 3 with a dominant T and there's 1 with no big letter. So it's going to be 3 to 1, and the percentage there is, since there's 3 of them, that's 75% dominant and 25% recessive. Alright, do one more practice here, and then you can try the last two on your own. A homozygous dominant, so there's two big letters. And so if white is dominant, then red has to be recessive. So that means the other letters are going to be little a, little a. So then we fill in our Punnett square, big A, big A. Again, I should use T's. Big A, big A, big A, big A, little a, little a, little a, little a. Alright, so we get our genotype percentage would be 100% big A, little a, because they're all big A, little a. These, because there's a big A in the box, there's a big A, little a, there's a big A in all the boxes, so our phenotype ratio would be 4 dominant to 0 recessive. So there'd be 100% dominant. Alright, so now we can apply these to our pedigree. So I'm going to skip the next two examples that are on your sheet. So far we've done everything but these two. And now we're going to go through this pedigree here just to save some time. Alright, the pedigree. So again, you know how to read a pedigree. These are carriers. They're big letter, little letter. These are little, little because it's recessive. So what we're going to do is find 1 and 2, the percentages of sickle cell trait, and no sickle cell. So let's look at 1 and 2. Well, your sheet doesn't have my letters on it. Alright, so 1 and 2 are up here, these two. Alright, so 1 is little r, little r. So we're going to make a Punnett square. Alright, so number 1 will be little, I'm going to use t's here just because it's easier. Little t, little t. Number 2, because it's not filled in at all, it's going to be big T, big T. Alright, so there's going to be a big T in all these boxes because they go all the way across. And then there's going to be a little t in all these boxes because they go all the way down. So all the boxes are going to be big T, little t. So if we go back to the sheet here, 1 and 2 percentages of sickle cell anemia. So there'd be 0% sickle cell anemia because they're all big T, little t. So all 0% little t for sickle cell anemia. It would be 0% for no sickle cell, which would be big T, big T. And then it would be 100% big T, little t, which is the sickle cell trait, the carrier. Alright, so we'll do one more here quick. So 3 and 4 are down here. So now we do our Punnett square. So number 3 is big T, big T. And number 4 is big T, little t. So you fill it in. And you're going to get, basically there's going to be 2 with big T, so 50% without the trait, or without any sickle cell. 2 with big T, little t, so 50% with the sickle cell trait, and then 0% for sickle cell. So it'd be 50% no sickle, 50% for the trait, and 0% for sickle cell. Alright, and then you can try the last one down there. And then email me if you have any questions.